We have a great program this morning, and we'll go ahead and get started with that. The first session is Stem Cells, Fact, Fiction, and Future. This is an exciting area that we are very interested in. We have Dr. Hank Paulson, and Hank and I were talking a while ago, and we have something in common. He was born in North Carolina, and I'm sure deep down in his soul he wants to return to North Carolina because he loves it so much. But I can't read all this fine print in his biography with his light up here, but he is currently at University of Michigan, where he is part of the ongoing group of neurologists and ataxia scientists, and also he will have his graduate student Lauren Moore up here with him as well to explain some of the things that are happening in the exciting world of stem cells. So, Dr. Paulson, if you want to stand, that'd be great. Oh, you're already up here.
Thank you very much, Ron. This is actually always one of the highlights of any year to come to the meeting, and this has been a tremendous meeting. Our Birds of a Feather session yesterday on a particular ataxia was super high energy with lots of attendance, and I learned a lot out of that, and thank you all for your participation in this meeting. It's also a tremendous thrill. It's not an opportunity you get very often to tag team a lecture with one of your own students. And so at some point in this talk, I will turn it over to Lauren Moore, who is a graduate student in the laboratory. Now, does this advance the slides? Whoops, like we jumped way ahead. So I guess I have to start with disclosures. I have received a research grant from a company on ataxia, Ionis Pharmaceuticals, and I have research grants from the NIH and the ALS Association, and in past years I've received grants, thankfully, from the National Ataxia Foundation. Lauren Moore has no specific grant relationships or business relationships. She does have a grant from Michigan Brain Initiative, and she'll tell you about other interesting relationships that relate to her presentation here today. So there's a lot of ground to cover. I know many of you wonder about stem cells. What are they, and can they be a therapy for my form of ataxia? We had a pretty good discussion yesterday in our own Birds of a Feather about this. But before we get into a discussion of can they be used for therapy, how are they being used currently, and how might they be used, I think we have to go through a couple background definitions. And I'll try to switch between the two sides. I'm a lefty, so I might go there more often, but we'll start on the right side. And so here is, what is a stem cell? Well, it's an undifferentiated cell from a multicellular organism that can give rise indefinitely to more cells of the same type, and from which certain other kinds of cells arise by differentiation. So here you have, and of course, the ultimate stem cell is a fertilized egg, right? I mean, that is something that is totipotent. It can make all kinds of tissues. And you may recall, some of you are old enough to recall the sheep Dolly, when Dolly was cloned years ago. You can, in certain situations, take a cell and allow it to go back to a de-differentiated state and then generate all kinds of tissues. So you can see in this picture here that this stem cell has generated a bunch of different types of tissues, including neurons. Well, there are a couple terms that we use when we talk about stem cells. You might see them thrown about by scientists all the time. iPSC, ESLs, pluripotent, totipotent. What does that mean? Well, stem cells can differentiate into many different types of cells. If they can produce all types of cells and even generate a full organism, then they are totipotent. They have the total potency. If they can produce many, but not all types of cells, they are called pluripotent or multipotent. And so you'll hear the term induced pluripotent stem cells often, or iPS cells, and those represent those type of cells. What are the sources of stem cells? Well, there are at least three that we'll talk about today. There are adult mesenchymal stem cells. These are found in many different tissues in our body. The bone marrow has mesenchymal stem cells. Fat has it. We heard yesterday about someone who underwent liposuction, had mesenchymal stem cells taken from his fat and injected back into his body. We don't know exactly what various roles they play, but among them are probably playing a role in normal tissue repair. And so many of the stem cell trials that have been going on out there now do involve these mesenchymal stem cells, either derived from the same person, autologous, or derived from some other person. There also are induced pluripotent stem cells. These are most often derived from skin cells, but you can also generate them from blood cells. And many of the scientists at the meeting that just ended yesterday have been using these iPS cells to study disease pathways and try to find medications or compounds that can help in disease processes. And then finally, embryonic stem cells. These are stem cells that do come from an early embryo, and they are truly totipotent cells. Here is a little more about embryonic stem cells. You can see here that you have, I'm sorry, I keep pushing the wrong button. You can see that we have a sperm and an egg coming together and generating the early blastocyst, which is a rim of cells and internal cells. These inside cells can be taken out and grown in a dish, and they can form clones or colonies of totipotent stem cells that then, under various conditions, can be differentiated to form skin cells, muscle cells, or neurons or brain cells, which we often want to study. There are limitations to the embryonic stem cells, and these include ethical considerations. This is not something that everyone agrees to, the idea of taking, for example, a fertilized embryo that might otherwise be discarded and generate cells or stem cell lines from that. There are, in this country, NIH-certified stem cell lines that can be used by American researchers using NIH grants. Some of you may recall that years ago, President George W. Bush made a decision that existing human embryonic stem cell lines could be used for research, but no others could be generated and used for research. Since then, President Obama released the rules and allowed for additional stem cell lines to be generated. So there are now a lot more stem cell lines, many of them disease-specific. That is, they've been derived from embryos that carry a particular disease mutation, including a number of ataxia such stem cell lines. And these are certified for use by American researchers with NIH grants. There are some countries in the world where you cannot do this, in Europe and elsewhere. So there are limitations to this, although the benefits include the fact that they are totipotent and can rapidly be generated into tissues. Okay, here's the iPS cells, what I mentioned. Again, you can take these from a skin cell, or you can take from a blood cell. And if you give a certain set of factors, these happen to be gene products, it can allow these differentiated cells to de-differentiate, take on essentially a stem cell configuration that then, with appropriate factors, can be turned into, again, different types of cells, including neurons. Great benefits here. You can take skins from anyone here and generate an iPS line. Scientists can take it from young people, old people. You can take it from a blood draw. So it's easy to do, and it's widely used now already by scientists around the world. There are some limitations. They are not totipotent. It's difficult sometimes to get them to differentiate into certain kinds of tissues. There is a lot of heterogeneity from line to line, and what scientists have learned over time is that you can't simply study one iPS line. You have to study several iPS lines, and you need to also generate the appropriate control lines that have the same genetic material if you're looking at how a particular mutation might affect things. So if you're looking, let's say you're looking at SCA1, which is a dominant ataxia, and you've got the mutation in a line, you're going to want to have the control line that's derived from the very same cell, but you've changed that mutation back to the normal configuration. And then mesenchymal stem cells. These come from all over the body. I'm just going to point out one thing. I've highlighted bone marrow here. Now, if you think about, have there been successes with stem cell therapy? I think the answer is a clearly unequivocal yes. Bone marrow transplant is essentially a stem cell therapy. People who have cancer will undergo procedures, radiation, or chemotherapy, or combination that essentially kills all of their immune system. Okay? And then placed back into that person are some of their own, essentially, stem cells from their bone marrow. and then it can reconstitute, or from someone else, a donor, and it can reconstitute the entire blood system in those individuals. So that's a clear success in stem cells. Now, that's not the brain. That's not a neurological disorder, but it shows you the power of a cell that can differentiate into different tissues and replenish something in a human being. But again, these can be taken from virtually any part of the body. There are many different areas where you can find mesenchymal stem cells. So what do stem cells have to do with ataxia? Well, number one, they're being investigated as a potential therapy for some of these neurological disorders. Number two, the stem cells and the study of stem cells can provide new insights into the disease process. Let's call this disease in a dish, that could lead to new approaches to therapy. And number three, they're a pretty good model system to test novel therapeutic strategies or screen potential compounds before moving into human clinical trials. Can they be used as a therapy for ataxia? Well, putting stem cells in the brain to restore lost neurons is, at this point, an unrealized dream. Now, we were talking yesterday in the Birds of a Feather. I think I had no concept that we would be able to generate induced pluripotent stem cells. We could take a skin cell and make a neuron out of it 20 years ago. It didn't seem conceivable. Now we do that. I had no concept 5 years ago that we could edit a gene very easily with simple molecular techniques. We can do that now. I had no idea about that. So dreams do get realized, and there are certainly possibilities, and we need to push forward to see how far we can go with stem cells in terms of understanding disease and developing therapies for disease. I think it's important, however, to note that to date there are no well-controlled trials that have shown benefit from stem cell injections into any form of ataxia in humans. There are some successes in mouse models, but as we all know, as much as we like Mickey, mice are not humans, okay? And there are limitations to trying to extrapolate from a mouse model to humans. Now, cell transplantation studies in Parkinson's disease, not necessarily stem cell, but cell transplantation therapies in Parkinson's disease have been going on for over 30 years. And they offer some hope for the idea that you could replenish cells or factors from cells in the brain of neurological disorders. But they also, I think, provide an important cautionary tale. The early claims for success in cell transplantation therapy in PD were not matched when placebo-controlled trials occurred in the 10 years following that. So there were very well-controlled trials that then did not show quite the robust improvements that occurred with cell transplantation therapy. It became clear, however, from these studies that you needed a large number of cells and the right kinds of neurons injected into the brain in order to have any kind of success. And I think it's very important to recognize there are individuals who received cell transplantations that had dopamine-producing neurons into their brain years ago and who now seem to still have some benefit from that. So, yes, there have been some successes there, although we're still a learning process. Parkinson's disease arguably is not as complex as ataxia. In Parkinson's disease, there's one population of neurons that die, and they produce the chemical dopamine. So if you can just get back in cells that make dopamine, and you can get the dopamine to the right spot, yeah, stem cell therapy for PD sounds really plausible. But it might be a little harder in ataxia. And the reason for that is that the network of neurons in the cerebellum is really complicated. Now, this is one type of neuron in the cerebellum. It's called the Purkinje cell. I'm sure many of you have seen pictures of this before. And it is a spectacularly beautiful neuron. Here's the cell body. These are what are called the dendrites. Huge branches, like a massive oak tree. This little tiny blip going down this way is the axon, the single output from the Purkinje cell. Well, why does it have such a big network? It has such a big network because it's integrating signals from a wide range of other neurons that are connected with it. Okay? So it's a really difficult problem. If you take a stem cell and stick it back into a cerebellum, will it make that kind of cell, and will it make the right kind of connections? If that's not complicated enough, if you look at the architecture of the cerebellum itself, so now here we're showing you this Purkinje cell Now it's red. So here's the same Purkinje cell now in red here, and here's its axon coming down. I think you can appreciate there are a lot of different cells of different colors in the cerebellum. We didn't know they had these colors, but here they are. You've got green, I think, granule cells, and you've got probably some basket cells here. You've got some other cell types here. All these little fibers going through the molecular layer are connecting up with the Purkinje cells. It may be a really tall order to ask stem cells stuck into the brain to reconstitute that incredibly complicated computer of neurons that make the cerebellum and that allow us to have control of motions. Okay? So, again, a tall order, but dreams have been realized before. I reviewed the literature over the last 10 years. There are over 35 clinical trial reports in the past 7 years on traumatic brain injury, spinal cord injury, ALS, stroke, multiple sclerosis, and spinocerebellar ataxia, including multiple system atrophy, a form of ataxia. And there have been over 35 such clinical trial reports in humans. They have been intravenous delivery of stem cells. They've been intrathecal, or that is to the spinal space, the fluid around the spine. Or in rare cases, they've been direct injections into the brain, or intraparenchymal. Most have used autologous cells, that is, cells derived from the very person who has them injected back into them. Nearly all of these have been open-label trials without placebo control. That's an important qualifier. Open-label trials have the risk of bias. If you don't have a placebo control, you can't account for the fact that the placebo effect is a huge issue. A very interesting issue that a placebo drug by itself can have a positive effect in many people. I think it is important to recognize that in these various studies, from my reading of them, the stem cell injections usually have been pretty well tolerated, and some of these open-label studies have shown some positive effects. I'm just going to give you one example from the literature. This is not something I had anything to do with. It's from Dongmei et al., Clinical Analysis of the Treatment of Spinocerebellar Ataxia and Multiple System Atrophy Cerebellar Type with Umbilical Cord Mesenchymal Stromal Cells. So they took, in this case, mesenchymal cells, not from the actual person, not autologous, but from someone else, and injected them intrathecally into the fluid around the spine 4 times in people who had SCA, dominant ataxia, or who had multiple system atrophy, cerebellar type. Then they did some measures. They measured their ataxia rating scales, and they followed up from 6 to 15 months. 10 of these individuals who received the injections remained stable for greater than half a year. That is, they didn't show a progression in symptoms. And 14 cases, so more than half, regressed to the pretreatment status, on average within 3 months. It's kind of promising, but there are important qualifiers. No open label. It was an open label trial. There was no placebo control. I don't think there were any carefully defined outcome measures, and it was a pretty heterogeneous or mixed group of research participants. So I'm not even sure they were powered to really show a statistically significant effect. So, you know, I'm a scientist and a neurologist, and so I want this to work, but I also want it to be done with great rigor and show me definitively that there's something worth pursuing. Now, I was one of many people who were the ataxiologists who provide consultation to the National Ataxia Foundation, who put together our statement on stem cell research and therapy. And I'm just going to read some of this. You can easily get it on the website. We are deeply concerned that some clinics are promising stem cell-based treatments for ataxia without oversight and other standard patient protections. They boast stunning rates of cures without scientific evidence to back those claims. In essence, the only thing they do provide is cruel health fraud at an exorbitant price, preying on the desperation that patients and families feel in the face of this untreatable neurologic disease. There is promise for stem cell therapies in some neurologic disease, but for now, patients need to know that currently there are no stem cells that can fix the brain, improve ataxia, or prevent the worsening of ataxia. Stem cells have great potential, but there are no shortcuts. We must use scientific principles that have been proven in the laboratory before we begin putting stem cells into people who are affected with ataxia. We must safeguard patients from unproven treatments that may cause serious harm. Our job as doctors is to do no harm. We want to advance the field. We definitely want therapies, but we also want to protect our patients when we have concerns about the operations of some of these clinics around the world. So, should the field of ataxia pursue stem cells as a potential therapy? I think absolutely. But It has to be done in carefully controlled trials with sound scientific basis based on compelling preclinical data. At this meeting, a company doing work in Taiwan, I believe, on a particular SCA, SCA2, has started a trial to see if mesenchymal stem cells can actually benefit those individuals. And I'm very pleased that they're using a placebo-controlled approach to the study. And so we'll keep our fingers crossed and hope that and other studies show positive results down the road. Well, what about the future? I think, in a sense, the future is now. We're using stem cells. We're using stem cells all the time to study disease processes. And what I'd like now to do is to turn the stage over to Ms. Lauren Moore, graduate student in my laboratory, who actually is using stem cells to try to understand disease process and screen compounds for a possible cure. After she's done, we'll come back on stage and answer questions. So I'll turn it over to Lauren.
Thanks for that introduction. My name is Lauren Moore, and I am a graduate student working in Hank Paulson's lab. And I study SCA3, one of the many ataxias. I do think that I am kind of unique among researchers in that I know what it's like to have one of these debilitating diseases in my family. My dad has tested positive for SCA3, and therefore I do have a 50% chance of also carrying the mutation. So like all of you, I have a very highly vested interest in finding a cure for this disease as soon as possible. And so first I just want to start off by giving some updates from the annual investigators meeting that occurred earlier this week. There was about 160 ataxia researchers who came and met and presented a lot of their new research on many of these diseases, including a lot of research that included stem cells. So one of the very compelling talks was entitled Transplantation of Neural Stem Cells as a Therapeutic Strategy in Machado's-Joseph's Disease, which is another name for SCA3. And in this study, they were able to create neural stem cells that were implanted directly into the mouse cerebellum. And they did see some rescue in some of the motor phenotypes that were in these SCA3 mice. But one of the things that the study presented was that it's unlikely that these cells were replacing cells that die in ataxia. It's more likely that they were having some kind of a nursing effect. So they were helping cells that were already sick and dying to maybe get a little bit better. And so this was compelling research, but it's very early. And so I'm very interested to see where this work continues in the future. A lot more of the talks, though, that included stem cells really focused on the ability to use stem cells as a model of ataxia in which we can learn more about this disease. And so that's what I'm going to talk about today. And that is kind of these rapid advances in stem cell technology, and that has really revolutionized the way that we study ataxia. And so to really understand how stem cells have advanced research in ataxias, I just want to present kind of how ataxias have been studied in the past. And so one of the traditional models of ataxia that scientists have used for over a couple decades are transgenic mouse models of these diseases. So these are mice that have the actual human mutation that causes ataxia put into the mouse DNA. So essentially you have a mouse with ataxia. And so these mice have enabled very invaluable insights into ataxia, into the processes that lead to disease, but they do have some limitations. As we all know, mice are not humans, and so therefore they have several limitations. For example, mice only live for about 1 to 2 years, and as we all know, many of these ataxias take several decades to develop. In addition, mice have a much simpler brain structure, and so we really can't capture all of the complexity of the human brain in these rodent models of disease. And also, many of these mice are inbred species, which means they have very little genetic variation. So that's kind of like trying to understand all of the ataxias by studying a single family. And so one of the results of these limitations is that many of the therapies that look very hopeful in mouse models of the disease have invariably failed in human clinical trials. And so we really need to find better models to study this disease in the hopes of finding treatments that are not only cure mice, but also can cure people. Another way that researchers study ataxias is through the use of a type of cell called a fibroblast. These are generally cells that are taken from, for example, skin biopsies. They can be taken directly from patients. So they are human cells that express the disease-causing mutation, but they're more like skin cells than they are brain cells. And as we know, ataxias mostly affect brain cells. And so this is kind of like trying to figure out what's wrong with a car engine by checking the tires. So the way that stem cells have really changed the way we study ataxias is that we can produce stem cells, human stem cells, and we can cause them to convert or differentiate into the type of cells that are affected in ataxias, primarily brain cells or, as they're called by scientists, neurons. So to give some examples of how stem cells have elucidated some new insights into the disease pathology and trying to understand some of the important steps that lead to cell death and dysfunction and disease, I'm going to present some of my recent findings that I've done while working with Hank Paulson up in Michigan. And so my research focus within the lab is to investigate causes of cell death and dysfunction in a specific type of ataxia called SCA3. And to do this, I've been using what I would argue is one of the best cellular models to study this disease, and that is the first SCA3 human embryonic stem cell line. This is the first and only in the world, and it was NIH approved in September of 2014. So it's been my prerogative to study this cell line and see what we can learn about ataxias from studying this cell, this stem cell line. And so before I can kind of show some of the new insights we've gotten into ataxia using this cell line, I want to first demonstrate that these cells really are ataxic and that they can actually replicate aspects of disease that are important to the human disease. So here in the top left corner, I have a picture taken with a microscope of an unaffected embryonic stem cell line. And I've outlined the outside of a single cell in red, and the inside, and then here outlined in white, is the cell nucleus, which is like the control center of the cell. And then I've also done the same for our SCA3 affected stem cell line in the bottom left-hand corner. And as you can see, there are some very apparent differences in these two cells. So one of the major differences that you can see is sorry, let me go back. Oops. So we've used a fluorescent marker to mark all of the ataxin-3, which is the disease-causing protein in SCA3 in green, and then we've marked the nucleus or cell control center in blue. And so as you can see, in the affected line, all of the ataxin-3 is almost entirely located within the nucleus, whereas in the unaffected cell, it's kind of located throughout the cell. And we know that this is an important step in the process of SCA3. When we look at either transgenic mouse models or post-mortem human brain tissue from SCA3 patients, this is one of the pathological hallmarks of the disease. In addition, we also see the formation of these really, really bright green, so these are highly dense centers of ataxin-3, and these are kind of like protein trash dumps within the cell or protein aggregates. So both of these two aspects, both the nuclear dislocation of ataxin-3 and the formation of these protein aggregates, are both known to be important and signs of SCA3. And therefore, we think that we can learn a lot about the disease from studying this line further. So of course, like I said, stem cells, their real power is that we can study the types of cells that are affected in human disease. And so using a number of different differentiation factors, we can take these undifferentiated stem cells, or the totipotent stem cells, as Hank discussed, and have them and convert them into human neurons that form very complex structures similar to the human brain, which is why we think of it as a disease in a dish or brain in a dish. And so looking at these neurons, we can really start to ask questions about what are the important steps in disease? What is causing these cells to die? What are some of the early steps that lead to cellular dysfunction? And so by comparing neurons that are derived from the SCA3 affected line versus the unaffected line, we can start to identify novel differences. And so even within the last couple of years, I've been able to identify some differences in these SCA3 neurons that were previously not mentioned within the literature. such as the formation of these vimentin-positive structures that you can see on the right in the SCA3 neurons. So in addition to advancing our understanding of SCA3 and ataxia toxicity, we can also use these SCA3 neurons to test novel therapies for ataxias. And so I'm going to talk a little bit about a new therapy that we've been investigating in the lab along with Ionis Pharmaceuticals and many different labs, which is called antisense oligonucleotides. So this is a new technology that is able to potentially stop genetic disorders by preventing the formation of the disease-causing toxic proteins. So this is called one of those gene silencing techniques. And so these antisense oligonucleotides, or ASOs, have been used previously in a number of different neurological disorders, and some have even advanced to human clinical trials. So one example that I'm going to highlight is a neurological genetic disorder called spinal muscular atrophy, or SMA. SMA is a disease, particularly of motor neurons, that causes progressive loss of muscle tone and muscle control. And this is a disease that primarily affects infants. And so many of these infants that are affected with this disorder do not live past 6 to 8 months. And so ASOs targeting the protein that causes SMA have led to very, very exciting results, including the fact that this therapy is safe and well tolerated in these infants. And also, many of these infants are showing significant increases in survival and motor abilities, and some of the participants are still alive 3 to 4 years later when they weren't expected to live past 6 to 8 months, which is very, very exciting. And due to these kind of early successes in other neurological genetic disorders, it's providing hope for many of the other genetic neurological disorders, such as ataxia. So ASOs have pushed forward with a number of other genetic diseases, such as Huntington's disease, ALS, and some genetic forms of Alzheimer's disease, which share many similarities with many of the ataxias. So we have gotten a hold of some of these ASOs, and we've been testing them in our transgenic mouse model of SCA3. And so what I'm showing on the left is, so after we treat these mice with the ASOs, we then look inside their brains and see how much of the toxic protein is present in each of these different brain regions, which I've highlighted here. The forebrain, which is kind of the front of the brain, the cerebellum, which is one of the main regions of the brain affected in ataxia, and brainstem, which is also highly affected in many of the ataxias, as well as the spinal cord. And so on the left, this vehicle is essentially untreated mice, or mice that are treated with a saline solution. And on the right is mice that have been treated with ASOs. And so we've just quantified the amount of protein in the treated mice versus the saline-treated mice. And as you can see, the amount of the toxic protein in these different brain regions is reduced to 25% or less in the untreated mice. And so we feel confident that this may be enough to have some prevention or even stopping the onset of ataxia in these mice. And so our next step is we really want to try to push these to human clinical trials as soon as possible. But as we've mentioned, many of the things that have proven successful in mouse models of the disease have failed in human clinical trials. And so this kind of creates a burden of trying to convince both the NIH and pharmaceutical companies that these will be successful in human trials, and we need to convince them to give us the money to do so. And so one of the ways that we can do this is by treating our SCA3-affected neurons. And so we're confident that if we can demonstrate that we can reduce the amount of the toxic protein that causes SCA3, as well as potentially reverse some of these disease-relevant changes that I've outlined already, that this would provide support for moving ASOs into human clinical trials. And the real hope is that we could potentially speed up the bench-to-bedside process in trying to move these from the laboratory to the clinics as soon as possible. And also, if we can demonstrate success in these stem cells, then we feel more confident that we will have success in patients. And so finally, I just wanted to outline a couple other uses of stem cells as a way to test novel therapies. So I've kind of talked about so far the use of ASOs as I would consider these as a candidate drug, where we already have something identified that we think will have success in preventing some of the changes in SCA3. But stem cells can also be used in high throughput screens. So there are these libraries of compounds and molecules that scientists have developed of FDA-approved drugs and drugs that we think may have benefits for different biological disorders. And so the use of stem cells can enable us to test tens of thousands to up to 200,000 molecules within a very short time, several weeks even, and see if any of these drugs that are already out on the market can be repurposed to treat ataxias. They also can be used by having induced pluripotent stem cells, which are cells that we actually take from a patient and reprogram into stem cells. We can have the hope in the future to have patient-specific therapies. And I'm sure that this is a very hopeful time for diseases like sporadic ataxias, where we may not know the specific genetic cause, but we could potentially take skin biopsies or some other cell sample from a patient and create neurons and study them in a very patient-specific manner. And this may, in the future, lead to the advent of personalized medicine. So this is a very, very exciting time in the ataxia field, and I do believe that we're very close to finding some cures for these diseases. And I just wanted to finish off by giving some insights that I've had as both an ataxia researcher and the proud daughter of an ataxia patient. But one of the first things that I realized when I joined the ataxia field is that there is no disease too rare. One of the reasons that I entered the field of ataxia research is when I started to learn about my family's disease, I thought, 1 in 50,000, how many people can really be researching this disease? And what I'm finding is that in some of these even rare disorders, it seems like there's more researchers than actual people affected with this disorder. So no matter how rare the disease there is someone out there working on it, and NIH is paying for it. And also, the ataxia research community is huge. This week, there was over 160 researchers, and each of those are just representatives from the lab. So many of these labs have more like 10 to 15 people, and not all of the labs are represented. So there are many people throughout the world. There are representatives from Portugal, from Australia, from Germany, from the UK. There are people all over the world working on this disease. And you do have advocates in the scientific community, and we really are focused on finding a cure. It may seem like when we talk we're interested in a molecular pathway or, this is interesting, these proteins interact, but the end result of all of these studies is the same. We all want a cure. New discoveries are happening every day. Like Hank said, the field has been revolutionized in 20 years, in the past 5 years, and even in the last 2 years since I joined the research community, I've seen changes within the field. I've seen new technologies. I've seen advances. And so it's really amazing the speed at which science is advancing right now. And I truly believe that cures are not only possible, but they're inevitable. And then finally, just to stick with the theme of this conference, there is a thin line between science and magic, and I am bewildered every day when I walk into the lab and get to see these cells up close and personal. So with that, I just want to thank you all for listening.